Thank you, Emily. Thank you very much. And thank you for coming this morning. Thanks so much for turning up to this incredible building. As Emily mentioned, I mean, the idea of these talks is to impart information, to tell us a little bit about what we're doing at the Crick, why we're doing it, why it's important. It's great to see some familiar voices here. It's even better to see lots of unfamiliar faces here. And I hope that today that it will be informative and perhaps funny as well on occasions. As I said, we're going to tell you really what the Crick is about, why we do what we do, and why what we do is important in our view. So it's a crash course on malaria. Unlike most courses, there's no tests at the end. In fact, you can test me, so don't worry about that as we go through things. So I'm going to divide my talk into really three sections. One, I'll tell you a little bit about malaria itself, the disease, why it's important. Secondly, how malaria is treated, prevented, and I hope that will be useful to you, actually, I mean, in a practical sense. And talk a little bit about drugs. Drugs are amazing things. You mustn't misquote me on that, it's really important, but they are incredible things. They're very difficult to make, and I want to talk about that quite a lot, actually. And I'm going to talk a little bit about rational drug development, how drugs are developed. And then I'm going to bring those two together and tell you a little bit about what my lab at the Crick does and why we do it. So those sort of three parts, really. So what's malaria? Well, before I start, I'll just talk about me for a second, a very short spell. Quite a long time ago now, I and my wife, Wendy, we went out to the Gambia, where malaria is endemic, and we worked there, or I worked there, at the Medical Research Council laboratories. So these were research labs working on malaria and many other diseases that are endemic to that part of the world. Those labs are now part of the London School of Hygiene and Tropical Medicine, just down the road. And it was run at the time by a wonderfully inspiring man called Sir Brian Greenwood, who is still at the London School. He still works there. And it was a fantastic place to work, a fantastic place to see how malaria impacts on people's lives in real life, if you like, from day to day. The MRC labs there did some wonderful work on malaria, the epidemiology of malaria, how it's spread, how it's transmitted, how people become immune, how they don't become immune. But essentially, if people are exposed to malaria from a very early age, they can become immune to clinical disease quite early in their life. But that's if you're exposed to it. If you're not exposed to it, as is the case, I'm sure, for most of us in this room, it's an extremely dangerous disease. So malaria kills lots of people. You may not be aware of this because it doesn't impact on our lives in the UK and in the global north from day to day most of the time. But just this slide really just covers a few figures. So it's caused by a single cell parasite, a protozoan parasite called Plasmodium. The most lethal form of the disease is caused by Plasmodium falciparum. And I'll talk about that quite a lot. You'll see the figures there. Lots of people die from malaria. There are lots of cases of malaria every year. And in 2022, these dates are a bit sort of behind the time because it takes time to compile the data. There are about 608,000 deaths worldwide. That's a lot of people, however you look at it. About 40% of the world's population live in areas where they're exposed to malaria. And so malaria deaths are responsible for about almost 3% of this damage. So they have a major effect on people. It has a major effect on people's capacity to work. And as a result, it's a great contributor to poverty. And even in the UK, and I put this figure in very specifically, people come back every year with malaria and die from it. There were 3 fatalities in 2021, but actually the figure on average is about 10 deaths per year. And I'll talk about that in a bit more detail, about why people come back to the UK, and even with the best clinical care, can die from malaria. So malaria is a killer disease, and it's not, there is this idea that you can, I'll talk about quinine a little bit, that quinine, which is, of course, as you know, is in tonic water, that malaria can be cured by a stiff gin and tonic. Well, you have to drink a lot of tonic water, about 10 litres or so. I'll come back to that, but it's a pretty important disease, a major disease. So what is it? What does it do to you? Well, This kind of summarizes up. So clinical malaria, everyone knows that it causes fever, and of course fever is a characteristic feature of the disease. But actually in its early stages, often people are not feverish, they're not febrile. But it can result in all of those complications I've put down there. Hypoglycemia, so that's very low glucose levels. Anemia, where you lose red cells. Kidney complications, brain complications, of course, which are probably the most severe form. Respiratory distress, comas, seizure, and all of these symptoms are caused by growth of the parasite in your red blood cells. I'm going to talk a lot about red blood cells, and as you all know, you have a lot of them. The most at-risk people, individuals, are children below the age of 5 and pregnant women, mostly in the first pregnancy. Now, it's easy to say this, but imagine if that were the case for COVID, during the COVID pandemic that we've all just been through. As we know, there were at-risk groups, but they didn't include children on the whole. If they had, it would have been a very different outcome. And it's caused, all these complications are caused by sequestration, so stickiness of malaria parasite infected red cells in your deep vasculature, so in the capillaries, the small capillaries of many organs in your body, your liver, your spleen, the lungs, and the brain, the placenta in pregnant women, and these lead to all the pathology of the disease. And malaria is a medical emergency. Even under the best conditions, as I've said, of clinical care, patients can deteriorate very rapidly indeed, within hours. So you've got to take it seriously if you think you've got malaria. It's been with us an awful long time. Malaria has been recorded in historical documents right back to in ancient Chinese writings. These are just a few examples from writings from the Mesopotamian Empire, which is modern-day Iraq and Syria. In Greek writings from Homer, there is even this idea that malaria was partly responsible for the breakdown, the fall of the Roman Empire. There is evidence of malaria epidemics in Rome, and actually a recent paper I have up here has identified malarial DNA in skeletal remains from the 1st and 2nd century AD. So it's been impacting on people for a long time. And I'm not going to really talk about this today, but it has altered the course of human evolution as well, most definitely. So for example, in malarial areas, we often see high incidences of genetic polymorphisms that partially protects against malaria, like sickle cell, for example. This is Oliver Cromwell's house in Ely, my favourite city. Cromwell allegedly died from malaria, or a complication of malaria. The story goes that he wouldn't take his quinine because quinine, I'll tell you a bit about this, quinine was associated with Jesuit missionaries. And as you know, Cromwell was a Puritan, a Protestant. He had a deep, skepticism about the Roman Catholic Church, and so he wouldn't take his quinine. As a result, he got ill and died. So, he sort of suffered from his own religious prejudices. But so malaria was very common in England, and in fact in the fenlands of Cambridgeshire and in the southern fenlands, transmitted by this mosquito called Anopheles atroparvus, which actually is still here. It's a very common mosquito throughout Britain and Europe. So with climate change, potentially malaria could come back. And there was in fact an outbreak in the UK in 1917 brought back by soldiers after the First World War. So malaria has had big effects on this country during the Second World War, the Vietnam War, the Korean War. The American Army lost lots of soldiers to malaria and many sort of sick days due to malaria. So it had a big impact on those wars. And in fact, the American Army has historically been quite a major contributor to malaria research, actually. So some good has come out of that. And the poster on the right is from a modern-day poster from Tanzania telling families what to do in case their children get sick and with fever. So what does it actually do? Well, malaria can be, if you like, divided into sort of 2 stages. It's the same disease, but one can morph into the other if you're not treated. So in its mild form, if you like, you get the fevers and the sweats, headache, confusion, even a sore throat, respiratory symptoms. Many of the symptoms that you see on the left-hand side are pretty non-specific. And this is why, if you come back from a malarial area, even if you've been taking your tablets, if you get symptoms that are flu-like, you should take them seriously and make sure that your GP is aware of that, because it could be the early stages of malaria. So myalgia is muscle pains, arthralgia is joint pains. All of these things on the left-hand side, they're pretty non-specific. And as you know, resemble many things that we come down with. And that can morph, if untreated, in the case of falciparum malaria, can morph very rapidly into all the stuff on the right-hand side, and we don't really want any of those. And as you see, they can be pretty serious, and you can go down very, very quickly indeed. So malaria is a serious disease. It's a medical emergency. And I just wanted to stress this because there is this idea, I think, in the sort of general media, that it's something that, you can treat relatively simply. And in fact, in the UK, it's a notifiable disease. So if you come down with malaria, and if it's diagnosed as malaria, it has to be notified to the public health authorities. And I just put this down really for your own usefulness. For a definitive diagnosis, blood samples have to go to the Malaria Reference Lab, which again is at the London School of Hygiene and Tropical Medicine. And you can get up-to-date information about areas that you may be visiting with that on that website there, and also advice on malaria prevention, which kind of drugs to take. Unfortunately, most antimalarials you can't get free on the NHS. You can't get them on prescription. You have to pay for them. But that's another story, really. Anyway, it's a serious disease and should be dealt with seriously. So, how do we prevent it? What do we do about it? So, before I tell you that, I want to tell you a little bit about the buggy that causes malaria. So, as I mentioned before, it's caused by the single-celled parasite, a protozoan parasite. It's transmitted by mosquitoes, Anopheles mosquitoes, and when the insect bites you, what it does is it injects a form called a sporozoite. Now, these are worm-like forms, they're very small, and these are injected into the skin, the dermis. And from there, they can very rapidly, they're motile, these things. They can move out of the dermis directly into blood vessels or into the lymph glands. And then they're really rapidly transported to the liver. In the liver, they invade your liver cells. Now, the important thing is that at this stage, you have no idea that you're infected. No idea. The first 10 days or so of an infection are completely clinically silent. No fever, no nothing. You have no idea. Which is why, if you're getting on the plane in Nairobi and you're bitten by a mosquito, the symptoms won't appear until a week or two later. Okay, so in the liver, the parasite then starts to divide. It invades a few liver cells, starts to divide, and each sporozoite turns into about 10,000 babies, 10,000 merozoites, these things are called. And these are then eventually released into the bloodstream. Again, you're still okay, you're still well, no fever, no nothing. And then they start to divide. They invade a red cell, they divide within that red cell, they burst the red cell open, and they invade a new red cell. And of course, each time this happens, you get more and more and more of your red cells infected. And over the course of several days, the proportion of red cells in your body starts to go up and up and up. And by the time you get to about 0.1% of your red cells infected, you'll start to feel ill. By the time it gets to 2%, you may be near death. So it doesn't take long once it gets to that stage of the infection. And so really, it's that stage of the infection that we tend to focus on, that so-called erythrocytic or red cell stage. Now, of course, that's not the only important stage in the parasite life cycle. And in fact, I'll tell you a little bit about this. The vaccines that we have at the moment target, in fact, the sporozoite, the stage that the mosquito injects. But nonetheless, disease is caused by this red cell cycle. And then these parasites can then form the occasional sexual form, they're called gametocytes. And these are the form that are taken up by another mosquito when it bites you, and then they go through a life cycle in the mosquito itself. Sadly, as far as we're aware, the malaria parasite doesn't make the mosquito ill. If it did, the world would probably be a better place. Anyway, so it just makes us ill, but not the vector. So that's how the life cycle of this thing works. And so it's quite complicated, as you see, but we tend to focus on this red cell cycle. So what we can do is we can see these things in red cells, and that thing on the right is what's called a Guim-sustained thin blood film. In fact, this is still used as the kind of definitive diagnosis of malaria. If you want to be really sure someone has malaria, they take a drop of your blood, they smear it out on a microscope slide, they stain it and look at it under the microscope. And you can see these individual parasites inside the red cells. And the parasites, the parasites are the little sort of purpley things inside the grey blobs. The grey blobs are your red blood cells. So you see the parasites are really small. Red cells are really small. And the bigger blob, that's actually a multinucleated form. That's a form where the parasites are divided inside the cell and are about to pop out and invade new cells. So how do we stop it? Well, an obvious thing is to stop the mosquitoes. And I should say, people have tried this for an awful long time. Of course, it does work. If you can prevent mosquitoes from breeding and surviving, if you get rid of sources, for example, of stagnant water, then you can very effectively reduce mosquito populations. And of course, in the end, if we're going to obliterate malaria from the world, we'd have to use a combination of approaches. And this is certainly one approach that is still being used. Quite a while ago now, it was decided by the WHO and others that one good way of doing this is to impregnate bed nets with insecticides. And this is an approach that has been incredibly effective. So insecticide impregnated bed nets, or ITNs, or long-lasting ITNs, which is the LLINS, these are very, very effective at stopping mosquitoes. Now, originally, and this was actually quite a serious concern, originally it was thought, I mentioned before that people in malaria endemic areas, they become immune. As they're infected, as they're exposed to malaria from a very young age, this constant exposure, as long as you don't die, this constant exposure can build a form of immunity. It's called naturally acquired immunity. And it was thought that if children were sleeping under bed nets, they might not get bitten enough to acquire that immunity. And this thing at the bottom in blue, this is actually an extract from a paper where they looked at the data retrospectively, and it became very clear that no, that isn't a problem. People do still become immune. So bed nets are a really good solution. The problem is insecticide resistant. Mosquitoes breed very rapidly, as I'm sure you all know, and they become rapidly resistant to insecticides. And things like pyrethroids, which are the insecticides that are used on bed nets, many mosquito populations are now resistant to these. Pyrethroids can be used in combination with other things, and in fact now there are new bed nets available where people are using combinations of insecticides, or insecticides combined with compounds that prevent the mosquitoes from breaking down the insecticides, if you like. And those are very effective as well. But essentially, insecticide resistance is a big issue. And, we all know that splashing insecticides around is not a great thing. They're usually quite nasty compounds. And they use, I mentioned here, IRS, which is this method for spraying the insides of houses with insecticide. Again, that's used widely, it has been used widely, it's been very effective, but again, the problem is insecticide resistance. So this is a real problem that people are having to deal with now. One of the best ideas that I came across recently, actually from a lab in the US, was rather than putting insecticides on mosquito nets, putting anti-malarial compounds on mosquito nets. So a group at Johns Hopkins showed that a drug called atovaquone, which is a very effective anti-malarial compound, if you put it on mosquito nets, the mosquitoes take it up through their feet, through their tarsal regions. And so you can kill the malaria parasite in the mosquito without killing the mosquito. And actually, this is a brilliant idea. I mean, they're following it up. And I think one reason why it's such a brilliant idea is that there are lots of compounds out there which we know kill the parasite, but aren't great drugs. In other words, you wouldn't put them into a human. But you could treat mosquitoes with them. So this is a great approach, which I think may be followed up in the future and could, solve a lot of problems though. Okay, so the other way of treating diseases, and we all know again from the pandemic, a really cost-effective way of dealing with infectious diseases and other diseases are vaccines. And people have been working on malaria vaccines for an awful long time, I can tell you that. I mean, certainly over the 40 years or so I've been in malaria research, people have been trying to make vaccines. It's been extremely difficult. Why? Well, it's very complicated. The malaria parasite is a complex organism. It's a parasite. There are no other vaccines against human parasitic diseases, to my knowledge. They have a complex life cycle. We've talked about this. They're big things. HIV has, I think, 9 genes. Flu has 11 genes, or is it the other way around? The malaria parasite has 5,300 genes, probably more. That doesn't mean that it's necessarily difficult to deal with it, but it's a complex organism, exists in many different, slightly different forms, and so creating a vaccine has lots of technical challenges. Anyway, it has been done. And there are now 2 vaccines that have been both licensed by the WHO. They're being rolled out in Africa. The phase 3 studies in Africa, in actually 3 African countries, were really successful. It's not the best vaccine in the world. It will get better, but it's going to save a lot of lives. And at the moment, it's looking really good. And I think, at the moment, it's not available in the UK, by the way, at the moment. It's been rolled out in Africa, which is where it's most needed, for sure. But, this is really difficult. If this works out, this will be a major success of modern science. And there are other approaches being looked at all the time. So, how do we mostly treat malaria? Well, as I say, the vaccine's there. By far the majority of treatments and methods used to prevent malaria rely on drugs. And there are lots of drugs. All these things on the left are anti-malarial drugs that exist, that have been developed over decades. I'll talk a bit more about them. You'll see that I've divided them on the right into 2 groups, prophylactic combinations, so those you would use to stop being infected, and those that are used to treat malaria, which are the lower lot. Most of the treatments these days rely on a drug called artemisinin and its derivatives. Artemisinin is I'll say a bit more about it, it's actually quite an old drug. It came from China originally, and has now been modified. So there are now these semi-synthetic versions called artemether, artesunate, etc. These are amazing drugs. These are fantastic drugs. But just like with the bed nets, microorganisms become resistant to things. We all know this. Anything that replicates quickly will become resistant to a drug or vaccine. And so there is resistance to many of these drugs on the left. Chloroquine, for example, is pretty useless against falciparum malaria over most of the world. Chloroquine, the 4th one down there, 5th one down there, is probably one of the best drugs of all time. It's cheap, it's easy to make, it's very effective, it's non-toxic, blah, blah, blah. But there's resistance nearly everywhere now. So in most cases, it will not be recommended as a drug. The big problem is resistance against these artemisinins. This has now appeared several years ago in Southeast Asia. It's spread now into East Africa. If artemisinin resistance becomes widespread across Africa, this will be a public health nightmare. This really would be bad. So everyone believes that we need new drugs. We have the vaccines, that's great. We have bed nets, but drugs are always going to be useful. And ideally, we need new drugs that work in new ways. And I'll explain why. So this is a poster that one of my colleagues used to put up years ago, a lot. Actually, it's no longer applicable really to malaria because, in fact, malaria used to be considered one of the neglected tropical diseases, NTDs as they're called, which encompasses things like schistosomiasis and leishmaniasis. In fact, now a lot of money has gone into malaria, and largely from some very wonderful rich people out there like Bill and Melinda Gates, for example, put huge amounts of money into malaria. And over the past few decades, the amount of funding for malaria research has really gone up. And that's how things like the vaccine has been supported. Anyway, the Medicines for Malaria venture, that was developed in 1999. It was a completely sort of new idea at the time, this idea of a public-private partnership of an organization funded by the World Health Organization, by the World Bank, working with pharmaceutical companies to develop medicines for malaria. And the reason why it was needed was because there's no money in malaria. There's no money in diseases that affect the poorer populations of the world on the whole. Of course, places like Africa and India also have large middle classes with lots of money, but I'm talking generally. So this MMV was a really important step forward, and it's now transformed how we develop new drugs against malaria, and they've got lots of drugs in the pipeline. So what about drugs? Going back to drugs, because they are wonderful things. So lots of our drugs are based on what are known as natural products, so compounds that exist in nature. Now, it's really important to remember that just because something is natural doesn't mean it's good. I mean, some of the most potent toxins known to humankind are natural. Phalloidin, botulism toxin, ricin, that's used on poison arrows. Lots of these things are natural, but they're really, really toxic, really toxic. In fact, phalloidin, I think, is one of the most toxic things out there. But a lot of our drugs derive from natural products. And I've already mentioned quinine. So quinine, on the top there, is derived from the bark of a tree called the cinchona tree. It grows in the high Andes of Peru. I mentioned the Jesuits and Oliver Cromwell. It's said that it was first brought back to Europe by Jesuit missionaries in South America, and is known of the Jesuits' powder as a result of that. So it's derived from an extract of the bark of this tree, and obviously was used in South America to treat fevers long before it ever came back to Europe. But it's quite a complicated, and any of you who are chemists out there would see that that's quite a complex compound to make, the top right. I mentioned artemisinin, this thing derived from Chinese ancient medicines. So this is from a plant called sweet wormwood, or Artemisia annua. Grows in very common around the world, and that compound there at the bottom is made from those plants. Again, a natural extract. The active compound artemisinin was eventually isolated by Chu Yu in 1972, and she won a Nobel Prize for that, because this is an amazing drug. However, making it is really hard. Natural compounds are often very difficult to reproduce in the lab, because in plants and in their natural forms, they're often created by enzymes that can differentiate between very subtly distinct forms of the same molecule, or isomers. So this is why these compounds are really difficult to make, but they're fantastic drugs, fantastic drugs. The problem is how you make similar drugs on a large scale, because obviously for a large population you need lots of these compounds. And so although these things are great starting points for drug discovery, they're often not very easy to mimic in the lab. Now, people have been able to do that with artemisinins. It took a lot of work. But actually, the other main point is that you don't necessarily need to know how a drug works to use it medically. And that's that sort of point down there. So artemisinins, as I say, were used for many years. Quinine was used for many years before people understood how it works. And in fact, we still don't know how quinine works, actually. And even artemisinin, there's some question about it. However, if you do understand how a drug works, if you really understand how a drug works, then it makes it a lot easier to make better drugs. It makes it a lot easier to deal with resistance if an organism becomes resistant to it, because you can make derivatives of that drug, or new versions that have the same function. So biological understanding, which now going back to what we do at the Crick, is really fundamental to drug development. It's not essential, but it really, really helps. And I want to give you a couple of examples of that, actually, before going back to malaria. So we all know about HIV, a terrible infectious disease caused by a retrovirus. I think, in fact, there was a talk in this series just a few weeks ago on HIV. In 1981, when HIV first appeared, at least on my radar, maybe it first appeared in the world, if you were infected with HIV, it was a death sentence. Your chances of survival were very, very poor indeed. Now, people can live with HIV and live effectively normal lives as a result of a wonderful, wonderful collection of drugs that have been produced to treat this disease. Now, you can't cure HIV. As yet, you can't get rid of the virus from a person's body, but you can suppress viral replication. The first drugs that appeared against, or at least within that first generation of drugs, were based on inhibitors of an enzyme, a viral enzyme called the protease. It turns out that what HIV does, what this virus does, is it makes its protein, when it infects cells, as all viruses do, it makes its proteins as long, what are called polyproteins. It then takes an enzyme and chops those proteins up to create the proteins that it needs, the scaffold that it needs to make new virus or new infectious virus. If you block the activity of that viral protease, you don't get infectious virus. You get virus, but it can't infect new cells. And that's exactly what people did. They realized this very early on. And so people working in labs on this protease, they made inhibitors and working with pharma companies, of course, pharmaceutical companies. In the end, they developed a whole bunch of inhibitors. And now, I've listed a few at the bottom. These are still in use, usually in combination with other drugs that hit other parts of the virus. But HIV protease inhibitors have been an incredible step forward. This is based on biological understanding of how the virus works, and basically came out of work from people who were just interested in this incredible virus and how it works. So these are rationally designed drugs targeting a specific biological target. Another great example, last one, hepatitis C. You may have heard of this. This is a virus that's been publicised quite widely as part of the infected blood scandal recently. Lots of people were infected with hepatitis C. In fact, hepatitis C has been around for a long time, and in fact, infects loads of people worldwide. I mean, over 300 million people infected. Epidemic in some parts of the world. It's associated with cancer, associated with liver cirrhosis, lots of nasty things. It's not a great virus to be infected with. 20 years ago, I'm sort of living in the past a bit. I would say 30 years ago, the idea of treating hepatitis C and clearing it from your body was completely unthinkable. No one would have thought that was possible. I mean, we actually knew friends who were infected with hepatitis C. And then again, people working in labs on the biology of hepatitis C realized that this virus works in a similar way to HIV. It makes these long proteins, it chops them up, it makes you viruses. If you inhibit the enzyme that chops up these polyproteins, you prevent viral replication. And these drugs, telaprevir and boceprevir, were the first drugs which were inhibitors of these proteases. They were licensed in 2011, and now hepatitis C is, in most cases, curable with these drugs. It's an incredible example of rational drug design based on understanding of how a microorganism, in this case a virus, works. Rational drug design does work. It's not easy, but it does work. Okay, back to malaria. So, what do we do? Well, I just wanted to talk about the guys in my lab here at the Crick. So these are the individuals who are in my group. And, labs in research institutes like this and in universities and in pharma companies, they essentially are very similar in their makeup. What you want is a bunch of people who are clever, ideally, who are thoughtful, ideally, who think about what they're doing and are really motivated, and who have different skill sets. And if you put all that together, as long as you all get on with each other, you can form a team that can be really productive and really sort of, in great detail work out how things work. So these were the people that I'm not going to talk about individuals in the lab right now, but in addition to that, we have collaborators, and that is the same for every lab in the Crick. We have funding bodies on the left. These are in some cases pharma companies like GSK, in some cases charities like the Wellcome Trust, which has huge amounts of money put it in for biomedical research. We have collaborators on the right there from all over the world, really, from different places, again with different skill sets that we can tap into and take advantage of, and vice versa. And at the Crick, we also have these things called science technology platforms, which are wonderful collections of people with very specialist expertise to help us do very specific tasks. Like, for example, to work out all the small molecules that are in a cell using metabolomics, for example. And this is one of the real strengths of the Crick, this sort of idea of having these STPs, as we call it, that works incredibly well. So what do we do in our lab? So we work on, I've already mentioned, this blood stage life cycle of the malaria parasite, because, as I say, this is what causes disease. So on the left here is a little video, which I think will loop, showing a malaria-infected red cell popping and releasing these merozoites, these invasive forms that go on to invade another cell. I think you can even see some invasion events there, where they bind to a red cell next to them and sort of go into that red cell. Invasion takes about 30 seconds. these organisms, once they leave one red cell and get into another, 30 seconds, really quick. And of course, once they're in a red cell, they divide again and repeat this whole process. The little statement at the bottom is from an ex-colleague of mine, John Williamson. I don't quite agree with that, but he was certainly right that if you know how to kill these things, it's a really great step forward. So we work on this blood stage life cycle. And this process of popping the cell, we call it egress. In fact, the same word is used to describe viruses coming out of a cell. And that was our particular interest. How do these parasites pop the red cell that they live in? And when we started this work, no one knew. So what we can do is we can culture malaria parasites in the lab. And that's really, really useful. We use human blood. That's why we take on PhD students. No, just joking. We use human blood. We get it from the blood transfusions, NHS BT as it's called these days. And we grow the malaria parasite in incubators in human red blood cells. Really good, really important. This thing on your right is an electron micrograph of a malaria-infected red cell. So this is taken down in a scanning electron microscope. And in this case, the red cell has been kind of split across. And you can see the parasites lying inside the cell, covered in a kind of duvet. The duvet is there's the red cell membrane. And then you see at the left, it says parasiferous vacuole membrane. That's a vacuole that the parasite lives within. When it goes into a cell, it actually forms a membrane-bound sac around it, and it lives within that sac within the red cell. And it digests the hemoglobin, the red protein of the red cell, in order to survive and grow. So, yeah, we can grow it, we can study it. And this is some work from Theo in the lab, who is very into robotics and AI. And he developed this automated system for culturing. Culturing these things is actually quite a lot of work. You've got to sort of change the medium and switch them around and transfer them from one flask to another. And he developed this system for doing it in an automated way, which of course is fabulous. Saves a lot of work. And the little graphs on the right just show that he could maintain the oxygen tension and the oxygen levels in these parasites. They need quite low levels of oxygen, and also maintain the temperature as well, quite well. So this was a great way, a great sort of help towards a lot of the stuff that we do. So how do they get out of a cell? How do they egress from a red cell? So again, this video on the left, which I think it will loop round and round. Now this is time-lapse, so it's speeded up. But I think if you look really carefully, you'll see that egress happens in two stages. So the inner bag breaks before the red cell membrane breaks. Now you might think, well, so what? Well, that's telling us something. It's telling us that these are two separate events, and that one happens before the other in time and space, and that's really important, as I hope I'll make clear. The other thing is, it isn't shown here, is that we could find quite early on that with certain drugs, with certain chemicals, we could block the breakage of the red cell membrane whilst the inner membrane would still break. And again, you sort of think, well, so what? Well, so what? That says that they're breaking through different mechanisms. There are different, that the parasite uses one way to break the inner membrane and one way to break the outer membrane. And that was kind of a first step in this sort of process. And then, over the last sort of 35 years or so, we've been studying this process. It takes an awful long time to work out how biology works. And what we've done over that time is gradually develop lots of quite clever, well, I use the word clever, I'm not being sort of quite clever and insightful ways to study these beasts. So for example, this video here, it's very difficult to show, but the parasites on the right are modified. What we've done is we've modified one of the genes in this parasite. And what you'll see is that they start to pop before the ones on the left. It's a subtle difference, but it's because the parasites on the right lack a gene that slows down this process. It slows down egress, so they come up more quickly. Now, I'm not going to talk in any more detail about that, but it gave us some very valuable information about how these things break the cell that they live within. We always knew that this was important. It turns out So I'm just going to summarize a bit of data now. It turns out this whole process is triggered by a protein, an enzyme called a kinase. Now kinases, we have several hundred kinases in our body that do lots of different things. They are very important in cancer because they tend to control how cells do things. They control reactions. And the blob on the right is actually an X-ray crystal structure of this particular kinase that the malaria parasite uses. This kinase triggers egress. Without it, they can't ever come out of a cell and they die. If you take drugs that inhibit this kinase, they can't ever come out of a cell and they die. So this kinase already is, in principle, a great drug target. Now, we and others, including our collaborator David Baker at the London School just down the road, have worked quite hard on this. We have compounds that inhibit this kinase. Unfortunately, they're not great drugs. And going back to what makes a great drug, for a great drug, for something you're going to put into your body, it needs not only to do its job, it's got to be non-toxic, it's got to have a reasonably good half-life in your blood, in your plasma, but not too long a half-life, otherwise it'll accumulate and then you might get ill. It's got to be non-carcinogenic, obviously. It's not going to have too many side effects. There are lots of requirements for a great drug, and it's one of the main reasons why drugs are so hard to make. Anyway, we know that this thing's a great drug target, so in the course of time, we hope that there will be good compounds that can be used as drugs against this. So, what happens? Well, it turns out that what happens is that there are at least two proteases, two proteolytic enzymes I've talked about with HIV and hepatitis C that are involved in egress. And what happens is that one of them is released from the parasite into the space surrounding it, and then it activates a second one, it chops it up, and then the inner membrane breaks, and then the outer membrane breaks, and the parasites escape. And these two enzymes, we call them sub1 and sera6, it doesn't really matter what they call. These are absolutely essential for the parasite to get out of its red cell. So I'm just going to say a little bit about red cells, because red cells are really important. We have lots of them. We take them for granted. But your red cells are incredible cells. I keep saying the word incredible. I've run out of an alternative. They last about 120 days if you're well. They go a long way in that time, about 300 miles, because they're whizzing around your body. They're very small, but they have this incredible flexibility that allows them to get through spaces that are smaller than the diameter of a red cell. In fact, they squeeze through the splenic slits in your spleen. They're incredibly deformable, strong cells. And of course, these are the cells that the malaria parasite lives in. Not many organisms live in red cells, by the way. The red cell membrane, the outer bit, is very flexible. And underneath that membrane is what's called a cytoskeleton. Many cells have a cytoskeleton, most cells have it. In the case of red cells, it's been incredibly well studied. And the reason why is because it's very easy to get lots of red cells. Simple as that. But it consists of this kind of, I often think of it as a chicken wire mesh, which comprises these nodes, these central bits, and then these flexible fibers that link them together. So almost like a hexagonal mesh. And this is absolutely essential to the strength and flexibility of red cells. And we know that because if people are born with mutations in their cytoskeleton, they often have anemia and problems with their red cells. Anyway, we know now that one of the things that the malaria parasite does is it clips this mesh. One of these proteases that it needs to get out of the cell clips the mesh, breaks it, and then as a result breaks the red cell membrane. And this thing on the left, it's called a Western blot, sort of just shows that. So that black line at the top, you'll notice that it turns into a double line as you go from left to right. And that just shows this protein being clipped. This protein is called beta spectrin. It's a very long protein. The parasite nibbles one end of it, it gets smaller, and the whole meshwork falls apart. So this is one means by which, so this is, if you like, an understanding for the first time as to how the parasite breaks down the red cell to get out of its cell. And these are just some little videos, again, of showing parasites that either lack or don't lack this enzyme. So the one at the top, so what we've got here is the grey blobs are the pictures of the parasite, and all the colours are different membranes of this cell, labelled in different ways. We label them with fluorescent dyes in this case. And see, at the top is a normal parasite, so it breaks down and eventually the parasites come out. The one in the middle, so the second one down, is a parasite in which one of these enzymes has been inhibited. In fact, it's gone. And you see what happens is it starts to break, it breaks the inner membrane, but the parasites never escape. And the one at the bottom lacks another one of these enzymes, and nothing happens. It just sort of sits there. It can't get out. It can't even break the inner membrane. So I said that the wrong way around, but anyway, so you basically see that, by getting rid of these enzymes, you end up with a parasite that can't escape from its host cell, and we know that these things just die. So if we can target these things with the right kind of drugs, we can kill the parasite. One of these enzymes we've studied in enormous detail, and this is an X-ray crystal structure of this enzyme on the left, and that's allowed us to learn a lot about how the enzyme recognizes the proteins that it binds to and cleaves. This is a really important part of modern biology. We can use this kind of information to almost in silico, if you like, on the computer, screen for drugs that may bind to this enzyme. In fact, this approach is only successful in some conditions, but in this case, we've actually managed to find inhibitors of this enzyme. So by using X-ray crystallography, which is something that we rely on using skills within the Crick, then we can learn an awful lot about how these enzymes actually work. And this is just some recent work where we've developed, with our collaborators actually in Latvia, developed drugs that are really effective at killing one of these enzymes. These are not drugs. These will not go into humans. They would be broken. They would probably have a very short half-life, but they're a good step forward. So it turns out that inhibiting any of these enzymes will probably, this just shows this sort of cascade of enzymes that we've worked out that is involved in getting the parasite out of its red cell. There are actually lots of them. There's PMX, there's PKG, CDPK5, SUB1 and SIRA6 and SIRA5. They're all involved. We know that inhibiting any one of them is likely to block the parasite from getting out of its red cell. And that's really what we're sort of focused on. And this, I think I've just got 2 slides left that really show that. This compound here on the left, this is a compound, it's called a nitrile. It's actually very similar to some compounds that have been used to treat coronavirus, actually. If we treat malaria parasites with this compound, what's shown here is actually 2 kind of different sets of malaria parasites. The ones at the bottom have not been treated, the ones at the top have been treated. And if I just show the next slide, by the time you get to 55 hours, you see the ones at the top are just stuck. They can't get out of their cell because we've inhibited the enzyme with this drug, whereas the ones at the bottom have come out and reinvaded new cells and continued their life cycle. So it is possible to develop drug-like compounds that target these things. Just the kind of approach that was used with HIV, with hepatitis C virus, and many other pathogens. So I'm going to stop there because I know I'm getting short of time. And this really just sort of summarizes what I said. I hope it's been sort of easy to follow most of this, but you can always ask me questions. So it's a single-cell parasite transmitted by mosquitoes. Only a few species of mosquito transmit malaria, by the way, relatively few. All the clinical symptoms are caused by this parasite proliferating in your red cells. You can block transmission with bed nets and with vector control, of course, but insecticide resistance is a big problem. There are some great drugs, there are some great anti-malaria drugs, but the problem is resistance. If resistance to artemisinin appear, that would be a really big issue. And we have been working to try and better understand the biology of this incredible organism. They are amazing. Doing science is a really great thing mostly because you discover how easy it is to be completely wrong in your intuition. You think, well, it's got to be this way, and then you find out you're totally wrong. And that's, I think, an approach that the scientific method really is very good at sort of revealing to you. And we've discovered in the end that invasion and egress involve these enzymes, these kinases and proteases, and we're working hard to find inhibitors of them. Anyway, I'll stop there. Thank you once again for coming today. It's been really good, and I hope that it's been informative and useful. Thank you.